Hi everyone, my name is Ferran Cruz and I am a graduate student at the University of Valencia in Spain. Today I will be talking about how climate and sexual selection drive interpopulation differences in the design of a UV signal in world lizards. It is generally acknowledged that chromatic signals can convey different types of information about senders. For example, they can warn about unpleasant taste or toxicity. They can inform about the sender's identity, allowing specific group or individual recognition. They can inform about variability or social luminance, or they can function in the context of reproduction as mate attraction or mate choice signals. In fact, one of the main challenges in the study of animal signals is assessing the type of information that they make available to receivers. Most studies of chromatic signal function focus on a single or a few populations, and many ignore potential interpopulation differences in signal design. However, this variation is important because it can provide clues about the selective forces shaping signal design and about the signal's information content. Often, interpopulation differences in signal design are attributed to differences in the signaling environment, and this signaling environment is mainly affected by variation in environmental contexts, including biotic and abiotic factors, and by social context too. For example, Manuel Leal and Leo Fleischmann found that the dewlaps of Anolis cristatellus of Puerto Rico varied in luminance between xeric habitats, where dewlaps were darker, and mesic habitats, where dewlaps were brighter, and they interpreted this variation as an adaptation to improve signal detection in these different environments. Here we focus on the European common wall lizard, Podarcis muralis. Males of this species exhibit a row of patches that we perceive as blue, but that actually have the reflectance peak in the near UV, around 360 nanometers. Previous work with this species has suggested that the UV blue patches act as a social signal informing receivers about males' fighting ability, but results are not conclusive and the available evidence is scarce. So our aim here is to study interpopulation differences in UV blue patches of Podarcis muralis in relation to the social and environmental context prevailing in each population. To characterize social context, we measure sexual size dimorphism, often used as a measure of sexual selection intensity. And to assess the environmental context effects, we measure climatic variables in each population. Between 2018 and 2020, we captured about 2,000 male wall lizards from 55 populations in the Eastern Pyrenees. And for each lizard, we measured as dependent variables the number and size of the UV blue patches. We measured the size as the surface area occupied by the UV blue patches relative to the total area of the scale row in which they are located. We also measured three spectrophotometric variables widely used in animal coloration studies: hue, which is the peak of maximum reflectance; luminance, which is the overall intensity of light reflected by the color patch; and chroma, which is a measure of the saturation or purity of that color. As explanatory variables, we used the degree of sexual dimorphism in each population in these three morphometric variables: the snout band length, and head width, measured as the mean of males divided by the mean of females. Also, for each population, we obtained 19 climatic variables from the World Clim data series, and with these variables, we ran a principal component analysis and we retain PC1 and PC2, which explain more than 80% of the variance. PC1 represents a gradient from hot and dry climates to cool and wet climates, while PC2 captures seasonality variation both in temperature and precipitation. Now to the results. First of all, we found variation between our 55 populations, both in social and environmental conditions. Here you can see, for example, the variation in body size sexual dimorphism. And our main result is that we found that these interpopulation differences in social and environmental contexts are associated with variation in signal design. On one hand, as you can see in these scatter plots, where each point represents a population, we found that the UV blue patches have more UV chroma in populations where males are larger than females, and more luminance in populations where males have larger heads than females. So UV blue patches have more chroma and luminance when sexual selection intensity is higher. This suggests that signal design varies according to sexual selection intensity, and therefore supports the idea that the UV blue patches are a sexually selected social signal. On the other hand, we also found a relationship between PC1 and both hue and luminance of the UV blue patches, with males in cool and wet localities having patches with less luminance and more hue. That is, more blue PS patches than those of males in hot and dry locations. This result suggests that there is local adaptation of signal design to the environmental conditions. And PC2 showed a correlation with the number and area of the UV blue patches and with luminance, with male lizards of localities with high climatic seasonality showing more UV blue patches and with less luminance, again suggesting some degree of local adaptation in this chromatic signal. To sum up, we have shown that signaling environment drives interpopulation differences in UV blue patches of Podarcis muralis, both via the social and environmental contexts. And these results agree with the assumption of this coloration acting as a social signal. And this study also highlights the importance of climate in shaping signals' local adaptations. Thank you all for listening.